Ever wonder what's really flowing through your bloodstream besides the caffeine from your morning coffee? Well, buckle up because we're about to take a roller coaster ride into the sweet science of blood glucose, aka blood sugar. The fuel that keeps us climbing those peaks so we can scream through those thrilling plunges. In this video, we're going to look at three things: why glucose matters, how we handle a sugar high, and how we handle a sugar low. First, we need to discuss what is blood glucose. Glucose is the simplest form of a sugar molecule belonging to a class of carbohydrates known as monosaccharides. Nearly all eaten carbohydrates, except for fiber, end up in the bloodstream as glucose. So it doesn't matter if you've eaten pasta, fruit, or candy. All these carbohydrate-rich foods are viewed by the body as sources of glucose for cells to use as potential energy. In particular, the central nervous system, red blood cells, and skeletal muscles working at moderate to high intensity are highly glucose dependent for their energy needs. It is important to note that glucose is used in the metabolic pathways of glycolysis to produce an energy-yielding compound known as adenosine triphosphate, or ATP for short. In other words, cells need to make energy from glucose to perform their job at their best. Let's get back to answering the question of how does the human body manage to distribute glucose throughout the body to every cell, ensuring a consistent supply, even between meals. This all comes down to understanding that the body attempts to maintain blood glucose ideally between about 70 to 100 milligrams per deciliter. This is the range that you would expect to see in someone who is in a fasted state in the absence of disease. Under normal physiological conditions, glucose can actually fluctuate from an ideal range of 50 to 140 milligrams per deciliter, depending upon the timing of meals, diet composition, and physical activity. What happens if we jump off the tracks of this homeostatic 70 to 100 milligram per deciliter fasting range? If we dip too low, the brain starves, leading to dizziness or even a coma. That's referred to as a hypoglycemic excursion. If we stay too high, the blood becomes syrupy, which causes osmotic diuresis, where glucose pulls water into the urine, resulting in excessive urination, subsequently leading to dehydration and long-term structural damage. This is referred to as a hyperglycemic excursion. In addition, if blood glucose is chronically elevated, that can lead to damaged blood vessels and organs like the heart, basically aiding the progression of cardiovascular disease. This is also a good time to mention the three apathies that can happen as a result of chronic hyperglycemia: retinopathy, which is eye damage leading to vision loss; neuropathy, which is damage to nerves; and nephropathy, which is kidney disease. The take-home message is that letting glucose run wild is like eating a second helping of dessert. Sweet at first, but it doesn't end well. High blood glucose levels can cause some serious damage when we don't regulate it properly. Let's illustrate how a typical day might look by graphing blood glucose on the y-axis and time over the course of 24 hours on the x-axis. These two lines represent our homeostatic goal of 70 to 100 milligrams per deciliter, while the lines in red represent the limits where symptoms may manifest. Upon waking in the morning, glucose might be closer to the 70 milligrams per deciliter mark. If we decide to have 2 buttermilk pancakes with syrup topped with a half a cup of blueberries, our blood glucose might increase to about 120 milligrams per deciliter. As the morning progresses towards lunch, we see a decrease in blood glucose to about 80 milligrams per deciliter. Once hunger and appetite cues signal us that it's time to eat that Philly cheesesteak sandwich you've been wanting to try, glucose increases once again. Perhaps the high fat content of the meal results in a gentle increase in blood glucose, with a gradual tapering of blood glucose to about 80 milligrams per deciliter. Then it's dinner time! Spaghetti and meatballs sound delicious, doesn't it? If not, just insert your own carbohydrate-rich food here as an example. As a result, you might expect blood glucose to reach about 130 milligrams per deciliter. After dinner, you decide to go for a walk, which results in helping reduce blood glucose concentration down to about 80 milligrams per deciliter. But wait! What would an evening be without a little fibrous snack like popcorn? which might increase blood glucose levels a little bit to 90 milligrams per deciliter. After retiring for the evening, blood glucose levels begin to slowly lower through the night, so we can do it all over again when we wake in the morning. What a roller coaster ride! But how do we make this tight control happen? That's where two very important hormones come into play. Let's start with insulin. The gastrointestinal tract is constantly on alert, detecting the presence of macronutrients and signaling their arrival through incretin hormones. These hormones act as messengers, relaying nutrient information to key organs like the pancreas to help coordinate the body's metabolic response. Specifically, when chyme enters the proximal portion of the duodenum, K cells release glucose-dependent insulinotropic polypeptide, or GIP for short, in the presence of carbohydrates and fat. GIP targets the pancreatic beta cells to secrete insulin into the bloodstream. Another hormone, glucagon like peptide 1, or GLP-1 for short, is released from the L cells in the ileum and colon owing to the presence of carbohydrates and fats. GLP-1 also targets the pancreatic beta cells to stimulate insulin secretion and suppresses glucagon release from pancreatic alpha cells. The combined end result is that these hormones help amplify insulin secretion, suppress glucagon, slow gastric emptying, and promote satiety, all of which help regulate blood glucose after ingesting meals. It is interesting to note that the class of medications known as incretin mimetics, such as semaglutide, tirzepatide, or liraglutide, are designed to mimic the action of GLP-1. In other words, by taking these medications, patients are magnifying the effects of bodily released incretin hormones, which have been shown to help patients regulate blood glucose, appetite, and body weight. To illustrate the coordination of insulin-lowering blood glucose level, let's use a diagram. On the left, we have the pancreas, which is responsible for releasing insulin into the bloodstream. Speaking of which, this red cylinder represents a blood vessel. On the right, we have a typical cell represented by this circle. As blood glucose levels increase, like after ingesting your mom's famous lasagna, concentrations will normally go over 100 milligrams per deciliter. In order for glucose to move from the bloodstream and into a cell, the hormone insulin must bind to an insulin-specific receptor located on that cell. I like to think of receptors as docking stations where the message can be relayed to the cell. Once insulin binds with the cellular receptor, this results in the translocation or movement of a taxi called GLUT4 transport protein from the interior of the cell to the periphery. In other words, insulin is sending a message to the cell to mobilize transporters to the outer edges of the cell to carry glucose into the cell. When GLUT4 pulls glucose into the cell, blood levels decrease and the cell is happy because it has glucose that it can use to make ATP. An interesting and useful tidbit in helping patients is that GLUT4 transport protein can be utilized as a taxi for glucose entry into the cell even without insulin binding to the cellular receptor. What? How is that possible? As it turns out, GLUT4 responds to a powerful stimulus that we should all be doing. Muscular contractions. That's right. When we go for a walk or engage in other physical activities, the same calcium released by the sarcoplasmic reticulum within muscle cells that enables muscle contraction to occur is also responsible for stimulating the movement of GLUT4 transport protein to the periphery of the cell in order to provide the working muscle with some glucose for ATP production. Therefore, exercise helps lower blood glucose levels. Awesome! One more reason to encourage regular exercise. You gotta love that. Okay, let's talk about glucagon. The pancreas constantly monitors blood glucose levels and responds by releasing the hormone glucagon as levels decrease. Glucagon circulating in the bloodstream binds to receptors on the liver that signal hepatocytes to increase glycogenolysis, or more simply put, the release of glucose from stored glycogen within liver cells. This newly released glucose then is pushed out to the bloodstream to help increase blood glucose levels. Let's use a graphic to visualize the coordination of these events as blood glucose drops below the 70 milligram per deciliter mark, also known as hypoglycemia. On the left, we have the pancreas, which is responsible for releasing glucagon into the bloodstream. Speaking of which, this red cylinder represents a blood vessel, so we can circulate things like hormones and nutrients. On the right, we have a typical cell represented by this circle. In the upper left-hand corner, we have the liver containing about 100 grams of glycogen. That's enough to supply us with about 12 to 24 hours of glucose in a post-meal state. Hours after a meal, glucose levels begin to fall, and in response, the alpha cells of the pancreas release glucagon into the bloodstream. As glucagon circulates in the bloodstream, this hormone eventually binds to G protein-coupled receptors on the liver that signal hepatocytes to increase glycogenolysis to release glucose. GLUT2 transport proteins allow glucose to move from a high concentration within liver cells to a lower concentration in the bloodstream. This results in an increase in the amount of blood glucose circulating so that a steady amount of glucose can be delivered throughout the body to provide cells with the substrate needed to make ATP. Of particular interest are the GLUT1 and GLUT3 transport proteins in the brain, which allow for a steady supply of glucose to neurons without the need for insulin. In other words, the central nervous system demands glucose and receives priority over the circulating levels of glucose despite there being low levels of circulating insulin. That makes for happy cerebral cells. Other cells relying on the GLUT4 transport protein like muscle and adipose tissue will just have to wait until the next delicious meal gets eaten. It's kind of like the shopping frenzy during the holiday season when parents scramble from store to store, determined to deliver that one prized gift to their child. They must succeed or there will be consequences. Just like a determined parent on the hunt for that special toy, the human body prioritizes the brain's need for glucose above all organs. If the brain doesn't get its toy, cognition, coordination, survival, etc., degrade as the glucose deficit increases. It's also noteworthy that epinephrine also results in liver glycogenolysis under stressful conditions like exercise and fasting. This added hormonal signal helps ensure a sufficient supply of glucose to cells in need. The approximate 400 grams of glycogen stored in muscle cannot be used to elevate blood glucose during hypoglycemia. It must come from outside of skeletal muscle. Whew! That's what I call a physiological workout. It's basically circuit training for the pancreas while we sit on a couch enjoying a delectable snack. The pancreas has to sprint with insulin to catch hyperglycemia, then powerlift with glucagon to pull glucose out of hypoglycemia. But wait, we don't let our pancreas do all the work. We can also favorably help by intentionally manipulating our diet. Let's take a closer look at this. The amount of carbohydrate we consume in one meal can directly influence the extent to which hyperglycemia occurs. That's what scientists refer to as the glycemic load. For example, consuming a carbohydrate-rich meal, like a slice of delicious New York pizza, will result in a higher total amount of glucose in the bloodstream. This results in a longer period of time needed to correct hyperglycemia back towards the homeostatic levels of 70 to 100 milligrams per deciliter. Diabetes educators actually refer to this phenomenon as the pizza effect. Catchy name, isn't it? The downside of prolonged hyperglycemia is that glucose molecules attach to proteins throughout the body that over time can lead to damage. This is a process called glycation that leads to advanced glycation end products, resulting in the three big apathies: retinopathy, which is eye damage leading to vision loss; neuropathy, which is damage to nerves; and nephropathy, which is kidney disease. And of course, let's not forget about cardiovascular disease. The type of carbohydrate consumed can have a profound effect on the rate of appearance of glucose into the bloodstream. Simple carbohydrates, like those found in sweet treats, can accelerate the entry of glucose into the bloodstream and cause large spikes of hyperglycemia. While more complex carbohydrates, such as those from rice or beans, take longer to digest and absorb, subsequently resulting in a much slower rate of appearance of glucose in the bloodstream. Moreover, if the carbohydrate consumed contains a higher amount of fiber, that can slow the rate of appearance of glucose even further, helping to blunt the hyperglycemic response to a carbohydrate-rich meal. But Dr. Santo, what about the other macronutrients, lipids and proteins? Do they have an effect? Absolutely! Mixed macronutrient meals serve to slow digestion and absorption, which subsequently results in a more weakened rate of appearance of glucose in the bloodstream and overall hyperglycemic response. So, eating a mixed macronutrient meal, choosing more complex carbohydrates, and moderating portion sizes all can help our body's ability to maintain a homeostatic level of glucose in the bloodstream with less effort under normal physiologic conditions. Wow! Blood glucose regulation is such a wild roller coaster ride with lots of nuanced twists and turns, right? It's important for healthcare practitioners to understand these mechanisms so that we can provide basic knowledge to our patients to help them master the management of their blood glucose, especially if they have diabetes. If you have any questions about this material, please reach out to your instructor. We are all here to help you position yourself for success. Have a healthy and productive day.